Hello dear students. Today we are going to learn about the development of human embryo during the second week of gestation. In the first week of gestation, after fertilization, zygote divides mitotically to form morula and a blastocyst then forms. Blastocyst has inner cell mass and outer cell mass. Inner cell mass forms the embryo proper or embryoblast, and outer cell mass forms the trophoblast, which forms placenta. The trophoblast cell layer helps in the implantation of the blastocyst in the uterine cavity, and in uterine cavity this is the endometrial layer, which is known as decidua, and the uterine stroma becomes more and more vascular, become more tortuous, and secrete more amount of mucus and glycogen. On day 8, implantation occurs. Implantation occurs with the help of syncytial trophoblast, which is a layer developed from the cytotrophoblast, and it has a common protoplasm, common cytoplasm with multiple nuclei. It forms after dissolution of cell membranes of the cytotrophoblast, and then a common mass of protoplasm forms, which is known as syncytial trophoblast. It has more fluidity, which helps in the invasion of the uterine wall by the implanted blastocyst. It helps in the penetration of the uterine wall to reach it to the uterine vessels, which are increased in the number due to the effect of progesterone, and these are the uterine glands, which becomes more and more tortuous, increase with the increased secretion of mucus and store more glycogen to nourish the developing embryo. Then cytotrophoblast, the trophoblast layer, which is the primitive placenta. Cytotrophoblast layer form of single cells with separate cell membranes and separate nuclei. It differentiate or forms further a syncytial trophoblast layer gradually, and these layers, these combination is known as trophoblast. And the inner cell mass form the epiblastic layer, which is a columnar cell layer. This is the columnar cell layer, this is the epiblast, this is the columnar, these are the columnar cells, these are the epiblast or columnar cell layer, and a hypoblast layer, a small a cell layer which is present below the epiblast, and this cell layer is known as hypoblast. It is of cuboidal cells, and the epiblast is of columnar cells. Epiblast forms a cavity which is known as amniotic cavity, and the hypoblast forms a cavity which is known as yolk sac. On day 9 to 10, the syncytial trophoblast forms the trophoblastic lacunae, which are the small cavities formed inside the syncytial trophoblast, and initially they are not filled with the blood, but they gradually enlarges and try to reach to the mother vessels of the mother, and when they reach to the maternal blood vessels or uterine vessels, then blood from those vessels pours into the these lacunae, and the cytotrophoblast start to form villi. A starts to form projections, which also further helps in the invasion of the uterine wall. And here we can see an amniotic cavity has formed, and here a primitive yolk sac is formed. This is the amniotic cavity and this is the primitive yolk sac. Primitive yolk sac is formed with the help of hypoblastic cell layer, and amniotic cavity forms with the help of epiblast layer. Now the zygote has completely implanted in the uterine wall, and at the site of implantation a fibrin coagulum has formed, and here this membrane which is lining the primitive yolk sac is also known as Husser's membrane. On day 11 and 12, uteroplacental circulation established. These lacunae reaches the maternal blood vessels or maternal sinusoids, and a uteroplacental circulation has established on day 11 or 12. And another cell layer which is secreted by this Husser's membrane, this layer is known as the mesoderm, extra embryonic mesoderm. This is the amniotic cavity and this is the primitive yolk sac. Now primitive yolk sac the linings which lines the primitive yolk sac starts to secrete a cell layer which is known as mesoderm. This mesoderm then start to cavitate, forms a cavity inside the mesoderm. This is mesoderm, and cavity starts to form inside this mesoderm, and it gradually, this cavity which is start to form here, these are the lacunae or cavity inside the mesoderm. This is known as extraembryonic cavity, and this cavity gradually merges to form a large extraembryonic cavity or chorionic cavity. And here is this embryo proper, which is attached to the trophoblast with the help of a connecting stalk, which forms of the mesoderm. And there is increase in the size of the villi which are trying to penetrate the syncytial trophoblast and try to reach the maternal blood vessels to help in the nourishment of the developing embryo. Here we can see the yolk sac is now known as secondary yolk sac because it is covered with the mesoderm, and this mesoderm which covers the embryo proper is extraembryonic splanchnic mesoderm, and this layer of mesoderm which covers which is near to the cytotrophoblast is known as somatic mesoderm or chorionic plate. Now the trophoblast has syncytial trophoblast. It has many nuclei with common cytoplasm, and it forms from the cytotrophoblast. A hormone secreted from this syncytial trophoblast is known as human chorionic gonadotropin. It is a glycoprotein. It has two subunits, alpha and beta, and it is secreted by the syncytial trophoblast. It helps in the maintenance of corpus luteum during the pregnancy, and it helps in the maintenance of the decidua. The syncytial trophoblast helps secrete the HCG, and HCG helps in the maintenance of the the corpus luteum. Corpus luteum secretes the progesterone, and progesterone helps in the maintenance of the early pregnancy by providing it support uh, the vascular and glandular uterine wall, which helps in the nourishment of embryo. Beta unit is detectable in the serum and urine around the second week of development, and HCG, this hormones doubles in the serum after the 48 hours of uh, measurement, and it peaks around 8 to 11 weeks of development. Low level can be seen in the ectopic pregnancy or miscarriages. High levels can be seen in molar or multiple pregnancies. False negative can be due to the hook effect, which is when the hormone level or HCG level are much higher than the antibody which can detect it. This is all about the development of human embryo in the second week of gestation. Thank you.